Hey everyone, welcome back to Self to Self. My name is Heem, that's Nick, and today we're going to be playing or quizzing ourselves and doing the last part of the gene therapy quiz, the hard level. Nick, how do
Yeah.
you feel about it?
I feel like you all should subscribe and like the video. That's important. But I'm not feeling, I'm gonna be honest, I'm not feeling too confident about this one. Last time, the intermediate one kind of kicked our butts, so the advanced one should be interesting. I will make sure that I leave the explanations up. I'm sorry I clicked through them really fast in the last quiz 2 weeks ago. That's my fault. But let's just get started. No more
Yeah,
we're not gonna walk along any longer.
yeah.
We're gonna do it. So let me go ahead and share the screen.
God, we haven't talked to too many people that have done gene therapy stuff, so it's this
I know
knowledge
it's
is purely our own from reading articles or whatnot.
right. There we go. Can you see my screen?
I can see your screen. Oh, so this is the advanced. Sorry, not I said hard.
Yeah,
It's the
advanced. All right, hey, let's
advanced.
get started. Is this good? Is like zoomed in and everything?
Yeah, that's perfect.
What cell type would not be a direct target for gene therapy? Red
Oh,
blood cell, muscle, liver, endothelium.
A,
Think so?
right? Because they don't have a nucleus,
Yeah.
so they
Yeah,
can't
yeah.
they can't really do mo
I like that.
I mean that
No, I
everything
like that.
else,
Yeah, I like that. Well, because let's think about it. Muscle, like let's say you were gonna re-engineer a viral vector, you would use like tetanus, right? For a liver, you would use Hep C. For the endothelium, I don't know, SARS. All right, I
I
like red
mean
blood
it's
cells.
saying direct target. So okay.
Boom.
So let's
Boom,
say
boom, boom. Hey,
yep.
1 for 1. Red blood cells are anucleated when mature, meaning they lack a cell nucleus. As a result, they have no DNA or machinery to translate code into protein. Beautiful, beautiful, beautiful. Next question.
Technically, doesn't translation happen outside the nucleus, like
Yeah.
in the
But
ribosomes?
they
So
don't have
like
any machinery. It doesn't have any like
tRNA, I mean,
ribosomes.
I don't know.
Doesn't have any ribosomes. Okay. Fetal hematopoietic stem cells used in gene therapy can be obtained from hematopoietic.
Fetal,
It's not the embryo and it's not the yolk sac. It's either the fetus or the placenta because hematopoietic stem cells are going to be for blood.
or like immune cells, not only blood.
Yes. Yeah. Well, white blood
I
cells and
would. Are you thinking placenta? Because I'm thinking placenta.
I'm also thinking placenta because you can harvest the umbilical cord blood and the
Yeah.
placenta. But okay, placenta. Let's go placenta. Let's go with our guts. You are wrong.
Yolk
The
sac?
answer is yolk sac. The yolk sac is the first site of hematopoiesis in a mammalian embryo. It functions as a development circulatory system for the human embryo. Before an internal circulation begins, fetal hematopoietic stem cells can therefore be obtained from the yolk sac. Huh? Really? Okay. I guess it wasn't. I guess we got confused maybe because it wasn't asking for it like what point. Because like when you say the fetus, I guess this is all in utero. Yeah, it's all in utero. Okay. Well, we would have gotten that one wrong anyways. I did select an answer at first. We got it wrong. Okay, next question. Which gene is often removed to create replication deficient adenoviruses? Oh Lord. What?
Replication deficient
Yeah,
adenoviruses? What does that mean to you?
I don't know.
So adenoviruses that cannot replicate. So it's not
Yeah,
viral. It's just like a delivery vessel.
yeah.
Dude, I don't know
E1.
why. E1, what are you thinking?
I was thinking L1.
You were thinking L1. I was looking at L1 too.
All right, let's do L1.
This is
Sorry,
this is this
people
is
who wanted
yeah.
to see.
This is a shot in the dark. E1.
Few
No.
transcription units in the early region, E1A and E1B, alter transcription regulation during infection and transformed by directly interacting with the cell proteins involved in the transcription and cell cycle regulation. You guys
Dang.
can do this. Okay, pause if you want to
That's
read it.
okay.
I don't care. That was stupid. Oh Lord, which gene is often inserted into an adenoviral vector to treat cancer by suicide gene therapy?
Ooh. Oh,
Oh,
not IL-2.
no, and I don't think it's
Okay.
VSVG
Why not?
because I think that it's A, or or it's C because I think that C is, I don't know what GM stands for. I can't remember, but I think that that's um no, I'm not gonna say because I could be something really stupid. Cancer suicide
You
factor? No, not
you
right.
should know this though.
But like TK would be tumor, like tumor killer, right?
Yeah, maybe if they name it that way. Um, this is like apoptosis, like whatever you think causes apoptosis.
My gut's saying C.
We can do that. And
It's
if
gonna
you're
be
right about A,
it's gonna be A. It's A. And it's because of thymidine kinase. I was wrong, right but wrong.
oh
Okay.
my God.
Enzymatic property that converts the drug against cyclovir into a toxic substance that can kill human dividing cells. Yeah. Against cyclovir, suicide gene therapy systems. Yeah. Okay. One for three, one for four. Okay. Which deficiency was attempted to be treated in the first patient who died during a gene therapy experiment? Oh, I know this one. It's this.
What
Jesse
is this one?
Gaussinger. Jesse Gaussinger died at the University of Pennsylvania in a treatment for this.
So
Yes.
ornithine transcarbamylase,
Yeah,
meaning
it's a it's a metabolic disease that you aren't able to break down um ammonia after because it's metabolic waste and it causes toxicity in like the brain and like usually kids die when they're like 5, but if you like take drugs and have a certain diet you can live. And Jesse was 18, had like a really bad episode. His doctor suggested that he goes and tries this clinical trial with this doctor at the University of Pennsylvania, and he dies because yeah, it's a whole ethical thing because he wasn't given the right form of consent. The researchers didn't report any adverse effects and like only reported his death. They didn't tell him that other people had like liver toxicity. He died from liver toxicity. They didn't know that. So whole ordeal, but it's that. Where's his name? First person died in clinical gene therapy trial suffered from deficiency. He was injected with adenovirus carrying a corrected gene in the hope that it would manufacture the needed enzyme. He died 4 days later, apparently having suffered a massive immune response. Having the viral vector used to transport, it was also liver toxicity. This led to multiple organ failure and brain death. Really sad. Really, really, really sad. Next question. How are viruses referred to in which their envelope proteins have been replaced by other proteins like VSVG to increase the tropism? Is it A, crippled viruses, B, replication deficient viruses, C, pseudotype viruses, or D, infectious viruses? I don't think it's D.
it's not B.
It's not B. I want to go with pseudotyped because we have like a pseudonym. Isn't that like same person, different name?
To increase tropism. What is tropism?
Is that cheating if I look it up? I'm looking it
Don't
up.
don't look it up. No,
Oh, okay.
no.
I want to go with C.
Um,
Do you want to go with A? Really?
A doesn't seem good. I C seems like I don't want to choose it, but it's the only one that like is left. I don't think it's B because B we
Well,
saw in the
okay,
other
fine.
question.
Let's think about tropism. When you think of trope, I hear like autotrophy,
Exactly. I think of like autotrophic or like things that
heterotrophy.
grow to increase trophism. It can't be crippled then.
I think it's pseudo. I think it's
Okay,
pseudotype.
let's try it. Oh wait. Oh, okay. Yes. Yes.
Viruses in which the envelope proteins have been replaced are referred to as pseudotype viruses. For example, a retroviral vector for use in gene therapy trials is the lentivirus simian immunodeficiency virus coated with the envelope proteins G protein from vesicular stromatis virus. Yeah, so they're basically just taking the envelope, adding a different type of viruses like outer coating so it can be more specific, but like an ABV might be better at delivering it or carrying the DNA and doesn't break down as easy, but it needs the keys to get into the cell. Okay. Number 7, which of the following viral vectors are most often used in clinical gene therapy trials?
I thought it's AAVs.
Yeah, I think it's AAVs. It's not lentiviral vectors. And like, yeah, see?
Yeah.
Yeah. Okay. Well, adenoviral vectors or adenoviruses. I feel like those aren't the same thing, but man, they really are
Hmm.
close. But whatever. All right.
Man.
Okay. They're in about 25% of all trials. Serotype 5 adenovirus are often used in gene therapy, in which the E1A and E1B regions have been deleted, which we learned is
Oh,
how.
they're deleted to prevent virus replication. I thought it
Yeah.
was like, oh yes, yes, yes. Yeah, yep, yep, yep.
All right. Ready for the next one?
Dang it, yep.
All right. Number 8, a 3-year-old boy was being treated for SCID using a modified retrovirus, but the boy developed leukemia-like condition. What appeared to have caused this? The disorder is known to cause leukemia in patients. The retrovirus inserted near a proto-oncogene promoter. The viral batch was contaminated with RCRs, and the retrovirus triggered a human T-cell leukemia virus. Okay, it's not D. Okay, I'm looking up what SCID is. I think that that's fair.
It's severe combined immunodeficiency disease. So it has something to do with your immune
Okay, so they were
cells.
they were treating a cancer
No,
immunodeficiency.
yeah, SCID is severe combined immunodeficiency. It's not a cancer.
Okay, well then I think
Yeah.
it's gonna be B.
Yeah, because if you're deficient, if this is a deficiency disease, the gene therapy is probably trying to do pro and grow stuff. So if it's inserted near proto-oncogene and then it just grows the oncogene, should be right.
All right, let's check. The
Yay!
leukemia-like condition of clonal lymphocyte proliferation was shown to be related to the retroviral vector integration near LMO2, proto-oncogene promoter, leading to activation and expression of LMO2. Okay, nice, 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 nice. So basically, for those who might not have taken genetics, you don't want to necessarily understand it, but like if you insert a gene too close to something like a proto-oncogene and you want that to
Wait,
replicate, or
so
no, it's downstream.
LMO2 is So wait, what exactly is LMO2?
Wait, so it's a gene
It's a promoter. LMO2 is
control.
a proto-oncogene promoter, and activating that leads to activation and expression of LMO2. Oh, so then if you activate it, then it goes towards like a leukemia-like condition.
Yeah, because it has a
Okay.
big role in the development of the hematopoietic system. So if like you produce too much of it, it's going to produce too many hematopoietic cells. Next. What is the general, what is in general not a risk factor in gene therapy using adenoviruses? Insertional mutagenesis, overexpression leading to an immune reaction, generation of replication competent adenoviruses, or toxicity due to the administered viral dose.
From the other questions, C seems like the answer to being because they take out the
Yeah.
E1, you know? So then
Yeah.
it's just like,
Like that's definitely a thing.
yeah,
Overexpression leading to an immune reaction. Yeah, because
yes.
it could be inserted in the wrong place. Toxicity due to administration. Absolutely, you can give too much.
Oh my God.
When adenoviruses infect a host cell, they introduce their DNA molecule into the host. The genetic material of the adenovirus is not incorporated into the host cell's genetic material. Oh, yeah, that's true. That's true. That's true. Lentiviral vectors are put into the DNA.
All
Last
right,
question.
we're
What is not a product name for a replication deficient adenoviral vector containing the P53 gene? This is a freaking shot in the
I
dark.
think A is correct. Gendicine, I think we had a question about that. No, 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 no. Like it is no, no. Like it is the product name. Like I'm saying Gendicine is the product name. So
Oh,
it's
okay,
either
okay.
Advexin, Oncorine, or SCH5. That just seems so odd. Like a product name as that,
Yeah. Ready?
I guess. Oncorine. Wow.
Okay. Introgen.
Shanghai Sunway Biotech. See,
All
we would have named that anything like
right.
that.
Your score. 4 out of 10. Hey, there's room for improvement, Heem. There's room for improvement.
Yes. Good job, Nick. We're so good. We
Yeah,
definitely
hey,
need to talk to some gene therapy people.
yes, for sure. We
They
definitely got
gotta educate us.
yeah, I guess. All right. Well, do you have anything else?
That's all from me, man.
All right. Well, this has been another episode of Sell to Sell. We want to say thank you for watching. If you made it this far, we appreciate you. And yeah, I'm Nick. That's Heem. See you in the next one.
Yeah.